Welcome to our weekly Next Chapter support meeting. We have William Yan from Duke University, and he is going to tell us about human brain mapping. And it's going to be fascinating. So thank you so much, William.
Thank you, Ms. Philel. Hi, everyone. My name is William. I'm a junior from Duke University studying biology. And today I'll be talking about human brain mapping. So why is it important? So the goal of a human brain mapping is to create a detailed map of the brain with different neuron connections and the different cells, glia cells around it to understand which regions are responsible for specific activities, behaviors, and functions. And its significance not only lies in diagnosis and treatment, but also understanding the mechanism of how these underlying um, functions perform. And it is also very important that many of the research nowadays, instead of using mice as a animal model, they would um, use tissue engineering to engineer something that's even more similar to human tissues to simulate the drugs on brain injuries. And a better understanding with human brain mapping would definitely make tissue engineering more accurate, as nowadays we don't even have a blueprint to engineer the brain on. So a very important project, and many nations are spending millions, billions of dollars trying to having trying to have a connectome or the mappings of the brain. So what are some of the current approaches? One of the approaches MRI, magnetic resonance imaging, which it uses magnetic field to take a picture of your brain. The advantage of that is that it gives a live image with relatively good resolution and provides longitudinal data. But in terms of scientific research, and especially trying to have a blueprint for tissue engineering, it still lacks cellular resolution. The second approach is called EEG, electroencephalography. That measures the electrical activity in different regions of the brain, and it gives very good temporal data as it records the electric signal from the brain regions lifetime with very good resolutions in terms of timing. But it is even worse than MRI in resolution, um, because how the procedure is performed, they will put different stickers on your skull, and it depends on the number of stickers. The most stickers you can put is definitely less than 100, and that will only give you 100 regions of the electric activity, which is definitely too little of a size compared to the billions of cells we have. And the third one is histology, which is by far the most accurate way of knowing what each individual cells or each proteins that's expressed in each individual cells. But the limitations of histology is that the imaging for a microscope requires thin slicings, which we call sectionings. And normally how a brain, after we harvest the brain, it needs to be dissected into thousands of sectionings, and that itself will take years of labor, let alone the imagings and the distortion of the image that comes with the mechanical stressings of physically cutting the brain open into 1,000 slices. So all of them have their limitations. And one of the solution is to create a transparent tissue, a transparent brain, and perform histology or stainings on that transparent tissue so that we can know everything that's going on in a three-dimensional model. Okay, so what are some challenges for this optical clearing? Well, one of the main challenges is that adult brain have a accumulation of very dense and sturdy molecules after decades of aged tissues. One of the things is that the extracellular matrix will get thicker and thicker. All of the collagens, the fibroblasts that's doing all of the connective tissues are going to become thick and dense, and that blocks the light from going through, which is why, and also with the lipids, which makes the brain looks white and reflect the light out rather than making it transparent. So one of the two main things that the scientists are trying to do is one is called detergent permeabilization, which and the goal of which is to try to digest all of the thick extracellular matrix proteins I just talked about. And two is the blood clot removal. As we know that after harvesting the brain, many blood clots will still remain, and that will give a red color rather than transparent. So how do they exactly do that? So first step is detergent permeabilization. The goal is to clear the densely packed extracellular matrix and ultrastructure matches without leaving behind a residual detergent. So the traditional method is just to use a SDS soap, which is a very common soap used in biological setting. What it does is that it forms a large micelle, which is also a bubble that that gets that will engulf the dirty stuffs, and ideally it will be pushed out by polar substances like water. But the problem is that the soap will get stuck at the tissue surfaces. So an analogy of this would be when we wash hands with soap, the soap is exactly doing the same thing. It's engulfing the fatty, greasy stuff from our hand using its non-polar side, while it also has a polar side, which allows water to take it away. But what happens in the traditional method is that the soap, for some reason, just keeps attached on our hand instead of getting those greasy stuff away. It just wraps those greasy stuff and the entire thing gets stuck on the hand. And honestly,
William,
that's not.
William, can I ask
Yes.
you a question? Are they doing
Hmm.
these experiments on a brain after someone has died? Or how do
Yes.
they? Okay, all right,
They actually performed it on a pig brain first before
okay,
moving on to a human sample.
okay.
Yes, great question. Yeah, so the traditional method does not work as the soap still is left on the sample surface. So they discovered a new detergent which uh with the abbreviation called CHAPS. It has a smaller size than traditional SDS soap, which means that deeper penetration. And it also has a higher critical micelle concentration, which means that you get more bubbles, you get more soap bubbles per millimeter of area. And this allows this new detergent to recline to the bilayer surface without leaving residues, and it's easier to be washed away by water and taking out all of the dirty stuffs. And the second is blood clot removal. So human organs carry some residual blood clots, and one of the very important functional group in blood is called heme, which is the picture showed on the right. And as we can see at the center of it is a iron molecule, which provides very strong autofluorescence and blocks light from traveling inside the brain. So one of the reagent that they find out is called N-methyl diethanolamine. And what this solvent does is that it carries the blood away and is easily dissolved in water so that um, with mixing this new reagent with water is going to wash out all of the blood clots.
And they're trying to do this because they want to make a 3D model of our brain mapping. So
Yes.
they're taking the brain, they're trying to clean it with soap, they're trying to get the blood clots out that would keep them from being able to see what was going on in the brain structure.
Yeah. The entire idea is to clear out all of these things that blocks light to make the brain transparent, and that allows better understanding of the brain structure. Okay, and last but not least is a process called delipidation, which by the meaning of the word means removal of lipid. They use a molecule called DCM, dichloromethane, for this delipidation process, and mainly because of three reasons. One is that DCM has strong ability to dissolve lipid. As we can see on the right, the structure is mostly nonpolar, and it has a low boiling point, which means that it minimizes the risk of thermal degradation. And thirdly, it's DCM selectively targets the lipid while leaving other components relatively undisturbed. So many of the case in brain or in any tissue, scientists tries to stain for proteins as those are catalytically active or are very important to cell functions. We definitely don't want something that while getting rid of lipid, also getting rid of those important proteins. So this selective specificity of lipids are very important, which is why it makes DCM such a good candidate for this delipidation process. So with these 3 steps, first clearing out all of the dirty, greasy stuffs, 2, clearing out all of the blood clots, and 3, clearing out all of the fatty lipids, the scientists are able to create transparent brains. Yeah, and thank you for listening. That's my presentation today.
Yeah.
Wow, thank you so much.